this way, in fact. So the macrophage will manage to captate an antigen from the tumor, which is specific from the tumor, and from the tumor cell, sorry. And then it will present it to the lymphocyte. And by presenting this antigen, the lymphocyte will be able to establish a specific immune answer toward the cancer cell. That's it. It can be oriented toward an inflammatory phenotype, which is called M1, and in an immunosuppressive phenotype, which is called M2. The thing that in the phenomenon of cancer, both of these phenotypes are expressed in the macrophage at different level, and one of the roles of the lab is to understand how that polarization is done. One of the answers to that is the role of TERS. TERS is produced by cancer cells, and then it penetrates. Yeah, indeed, it penetrates the macrophage, and it activates several proteins inside of the endoplasmic reticulum of the macrophage, which are implied in the process of UPR, unfolded protein response.
Okay.
And this is supposed to deal with an external stress of the cell. If it's not one of these two proteins, it can only be the third one. What I want to do is to plate the macrophages in a 6-well plate. In fact, in 2 6-well plates, because I want to do duplicates for the results to be significative. And what I put in the first column, what I put in the first line is the vehicle, so nothing, just the growth medium that I put right now.
Yeah.
And in the second line, it's TERS. So the vehicle is my negative control, and TERS is the condition I want to test.
Yeah.
And then, since I want to study the effect of TERS on the different arm of the ER stress,
Yeah.
This process is called UPR, by the way, didn't tell you, unfolded protein response. So which is made of the PERK pathway that I showed you, IRE1 alpha pathway, and ATF6 pathway. So the IRE1 alpha pathway is studied by an inhibitor that is called FORMU8C.
Yeah.
So when there is no more, so when I add FORMU8C, it studies what happens in the macrophages with TERS or just the negative control when IRE1 alpha cannot do its role anymore.
Yeah.
And so if there is a different action in comparison with the negative control,
yeah,
it means that IRE1 alpha has an important role.
Okay.
In this column, I just put nothing. So
Okay.
it's another negative control,
Okay.
both for the growth medium and for TERS.
Okay.
So this is TERS alone, this is the growth medium alone.
Okay.
And then the third one, I put an inhibitor of the PERK pathway,
Okay.
which is GSK. If there is no difference with FORMU8C and GSK,
Okay.
it means that ATF6 is the pathway that is implied for TERS. This is what I will do. I will put TERS, FORMU8C, GSK in each well.
Okay.
And then I will wait 24 hours. So this is what I will do tomorrow.
Okay.
And then on Monday, 24 hours later, I will aspirate the growth medium of all of these wells.
Okay.
I will wash it with PBS.
Okay.
So only the cells will remain.
Okay.
I will aspirate the PBS again, and I will put a solution to I will put a solution to extract the ribonucleic acids.
Okay.
I'm going to isolate specifically the mRNA.
Okay.
And after I have isolated this mRNA, I will of course do a measurement of all the mRNA I have per sample,
Okay.
because it requires a minimum it requires a minimum of mRNA to do the next experiments.
Okay.
As I failed before because I didn't have enough. Then this mRNA needs to be transcribed into cDNA, what we call cDNA.
Okay.
So the thing that we cannot study RNA alone, because I don't know if you remember, but RNA has different nucleic acids than DNA.
Yeah, they have uracil.
Sorry?
Uracil?
Exactly, uracil. We want to convert it into thymidine. So we have to transform it into cDNA with an enzyme which is thermosensible.
Okay.
No, which is I don't. Yeah, sorry, I don't remember. It's a reverse polymerase, like the one that you have in the HIV virus.
Okay.
And then once I have my cDNA, I can put specific probes of the genes I want to study.
Okay.
And then after that I will put the probes, and I will put the whole cDNA plus the solution in a machine that is called qPCR machine, and I will be able to have the relative level of all the mRNA I'm looking for each sample. And then I will be able to compare if one sample has a higher expression of certain gene than one another.
Okay.
That's it. And then if I manage to get all these results, because I failed twice before,
Okay.
so if this time I manage to do it, I will be able finally to have my fifth figure for my report. If you have a box, and this box is split in half with the membrane, that just lets the water passes. Then you put a protein on one side of the well, but not the other. Then the water from one side will start to go through the membrane to equilibrate to try to equilibrate the concentration of the protein. And the thing that the osmolarity of the blood in the osmolarity of the solution in the cells of the body is of 300 micromolar. 300 micromolar of salt. If you put too much salt at the exterior of the cell, it will burst because it will absorb all the. No, sorry. If you put too much salt outside of the water, the cell will shrink because all the water of the cell will go out to equilibrate the concentration of salt that is too high. And if in the contrary you just put water, the cell will die because it will burst. All the water outside will go into the cell to equilibrate the concentration of salt. And this is why you always need to add. You always need to have an osmolar solution when you do injections, because if you just inject water inside the vein of someone, he will faint.